Biomedicine joint crash course with the RCSB Protein Data Bank. My name is Stephen Burley. I'm the director of the RCSB PDB with appointments at both Rutgers and UCSD. I'm also the founding director of the Rutgers Institute for Quantitative Biomedicine, which is hosting this event. We are here today to provide you with a crash course in working with the new rcsb.org research-focused website. The crash course is being recorded. The recording will be posted on our YouTube channel within the next 24 hours. And the presentation PDFs will be available to any of the participants who are kind enough to complete the exit survey for us. So before introducing the agenda, I just wanted to be clear as to what we mean by a computed structure model. This graphic taken from a recent New England Journal of Medicine article contrasts the steps involved in experimental structure determination with either X-ray crystallography, cryo-electron microscopy, or nuclear magnetic resonance spectroscopy, resulting in a well-validated structure, which experimental structure that's often only part of the polypeptide chain, but has minimal low or very low confidence regions. Functional ligands are often present, such as the image depicted here of a protein RNA complex. This information goes into the protein data bank and is then publicly disseminated through either our website, rcsb.org, or one of our worldwide protein data bank partner websites. Contrast this with what we mean by computed structure model, which are those predicted structures coming from artificial intelligence, machine learning methods that take advantage of both what's known about protein structures in the PDB and genomic data pertaining to the protein of interest or closely related proteins. And it's the amalgam of this information and an iterative process about which you will hear more that allows for generation using these AI machine learning methods of a computed structure model. These are typically full-length polypeptide chains. Particularly for eukaryotic proteins, they may include substantial numbers of either low or very low confidence regions that are probably disordered, and no functional ligands are present. These are being publicly disseminated through AlphaFold DB and the Model Archive and other resources. The motivation, the rationale of the RCSB Protein Data Bank for including computed structure models is enumerated here on this slide. They provide structure data for all model organism proteomes, which is not the case for many of the proteomes that are only partially represented in the protein data bank. For example, the proteome of Arabidopsis thaliana has about 5% coverage in the protein data bank. Addition of computed structure models for all of the Arabidopsis thaliana proteins brings that number close to 100%. Unlike many structures in the PDB, computed structure models cover entire polypeptide chains. It is it's the case that now that computed structure models can be delivered alongside the experimental structures in the PDB using the full functionality of the rcsb.org website for searching, analyses, and visualization, and you'll see this this afternoon. And computed structure models can be clearly identified as to their provenance and accuracy, and contrasting the provenance and accuracy of experimental structures represented in the protein data bank. The overarching goal of the RCSB PDB in updating and extending its data delivery capacities over the last few months is to provide better service for the more than 99% of our web portal users who are not experts in structural biology, and frequently find themselves frustrated when they come to the protein data bank and realize that there is no experimental structure available for their protein of interest from a particular model organism. Today's agenda is depicted here. We will be starting off with Brinda Vallat, and each of the speakers will successively introduce the other. And then I'll come back in at the end to provide some concluding remarks and also give you some information about approaches for sending us feedback. We are particularly interested in receiving constructive negative feedback because the only way we can get better is to understand how we may be only meeting the needs of our users in some partial way. So Brinda Vallat is a software developer here at RCSB Protein Data Bank. Her particular focus is on integrative hybrid structures and on the data model that is used for describing computed structure models with the model CIF data standard. You'll hear more about this. This is related to the PDBX MMCIF model data standard, which is used to describe all of the experimental structures in the PDB. So without further ado, I'm going to go off microphone, off screen, stop, and off camera, stop sharing my screen, and Dr. Brinda Vallat will take over. Thank you, Brinda.
Thank you, Stephen. I'm happy to give the introductory talk at this virtual crash course on exploring computed structure models at rcsb.org. Proteins are macromolecules made up of amino acids that determine their 3D shape. These amino acids interact locally and in long range to form secondary structures and folded tertiary structures. The folded proteins can also interact with one another to form quaternary associations that enable function. Why is structure important? Structure is important because it determines function. The folded structure of a protein is unique and stable and enables the protein to carry out its function. Protein Data Bank is the single global repository for 3D structures of macromolecules and their complexes, determined experimentally using X-ray crystallography, NMR spectroscopy, and 3-dimensional electron microscopy. PDB was established in 1971 and is the first open access digital resource in all of biological sciences. PDB provides open access to about 200,000 experimental structures with no limitations on usage. And PDB structures are expertly curated and validated. Although there are billions of proteins in nature, experimental structures are only available for a few 100,000. Experimental structure determination is often time-consuming, expensive, and can be technically challenging. Given that the sequence of amino acid residues in a protein chain determines its functional 3D structure, the question is, can we computationally model the structure of a protein from its sequence? There are two approaches for protein structure prediction. The first one is template-based structure prediction, also known as homology modeling or comparative modeling. This involves modeling the structure of an unknown protein sequence, known as a target, based on the similarity of the sequence to the sequence of another protein with known structure, also known as the template. Homology modeling has been successful when template structures with more than 40% sequence identity are available. The second approach is template-free structure prediction, also known as ab initio modeling or de novo modeling. This is applied when reliable structural templates are not available for the protein of interest. Template-free structure prediction usually involves large-scale conformational search to identify the lowest energy conformation. In recent times, intramolecular contact predictions based on coevolution data have been successfully applied for template-free structure prediction. Amino acid residues that evolve together are identified from multiple sequence alignments, and predicted to be in contact with each other in the 3D structure. And this information is used for structure modeling. To answer the question in the title of my talk, computer structure models are predicted models obtained using template-based or template-free modeling methods. I would like to quickly touch upon two community-led blind challenges that have fostered the development of both template-free and template-based structure prediction methods over the years. The first one is the critical assessment of techniques for protein structure prediction, known as CASP, and the second one is the continuous automated model evaluation, also known as CAMEO. In both cases, modelers predict and submit the 3D structure of a protein whose experimentally derived structure is yet to be released to the public. These models are then compared to the experimental structures upon release to evaluate both the models and the prediction methods. CASP is conducted every 2 years and CAMEO is conducted weekly based on pre-release sequences from the PDB. Both CASP and CAMEO have contributed to the evaluation and development of structure prediction methods over the years, and both challenges have relied on coordination with the structural biology community and the PDB. In recent CASP challenges conducted in 2018 and 2020, AlphaFold and its successor AlphaFold 2 were highly successful. This remarkable achievement was made possible by recent breakthroughs in applying artificial intelligence approaches and machine learning methods to structure prediction. What is AlphaFold 2? AlphaFold 2 is an AI system developed by Google DeepMind that predicts a protein's 3D structure from its amino acid sequence. AlphaFold 2 achieves accuracy comparable to low-resolution experimental structures. The AI system was trained by showing it the sequences and structures of around 100,000 known proteins from the PDB. So PDB data is the primary input for training the AI system. Without the enormous wealth of expertly curated and validated 3D structure data from the PDB, the application of machine learning techniques to train the AlphaFold AI system would not have been possible. AlphaFold models typically look like beads on a string, as shown here, with the beads corresponding to compactly folded regions of reasonably high confidence, and the strings corresponding to regions of low confidence connecting these folded domains. This is an overview of the AlphaFold 2 neural network model architecture, where several methodological advances were introduced that turned out to be successful. The input to the system is the protein sequence, and the output is the 3D coordinates. The training starts with a multiple sequence alignment rather than more processed features derived from these multiple sequence alignments. Information at the one-dimensional sequence level and the two-dimensional contact map level are iteratively transformed and exchanged back and forth in the network. The three-dimensional atomic coordinates are directly refined rather than the pairwise distances. And finally, end-to-end learning is employed where all network parameters are optimized by backpropagation from the 3D atomic coordinates through all layers of the network back to the input sequence. This slide highlights the performance of AlphaFold 2 in CASP14. Two examples of CASP14 protein targets in the template-free modeling category are shown. The first one is RNA polymerase domain, and the second one is the adenosine tip. The experimental structure is shown in green, superposed with the computational prediction in blue. As you can see, the computed model is very close to the experimental structure, and the carbon alpha RMSD is 1.14 angstrom in the RNA polymerase domain case, and it is 0.82 angstrom in case of the adenosine tip. So AlphaFold 2 can predict accurate structures as measured against the experimental result. AlphaFold 2 also provides two types of intrinsic model accuracy estimates. The first one is the predicted local distance difference test, PLDDT, which is a per residue measure of local confidence on a scale from 0 to 100. The second is the predicted aligned error, PAE, which is the expected position error at residue X when the predicted and true structures are aligned on residue Y. My colleague Chenghua will delve into the details of these model quality metrics later in his talk. AlphaFold protein structure database provides open access to AlphaFold predictions. In 2022, AlphaFold DB was expanded to include over 200 million protein structure predictions involving a broad structural coverage of sequences in UniProt. Following the success of AlphaFold 2 in the Cas14 challenge, Howard Hughes Medical Institute investigator David Baker and his team at the University of Washington developed Rosetta Fold. Rosetta Fold also uses an AI system similar to that of AlphaFold, but the network architecture used for machine learning is different from that of AlphaFold 2. The prediction accuracies of Rosetta Fold approach that of AlphaFold 2. Rosetta Fold has been applied in combination with AlphaFold 2 to predict the complex structures of core eukaryotic protein complexes. These structures are archived and freely available from the model archive at the Swiss Institute of Bioinformatics. Applications of computer structure models. Computer structure models are being used by structural biologists to accelerate experimental structure determination. These models are also used by molecular and cell biologists to develop testable hypotheses regarding proteins that are not present in the PDB, such as the switchgrass proteins, for which there are only 4 structures in the PDB. These models are also used to advance integrative structural biology studies, where predicted structures of individual proteins are fit into experimental maps of higher order structures to build the 3D structures of complex assemblies. This summer, we released an updated RCSB.org web portal, which now delivers over 1 million computer structure models together with approximately 200,000 experimental structures. RCSB.org provides an extensive set of tools and services which can be applied to search, analyze, and visualize computer structure models alongside experimental structures. These models are consistently distinguished from experimental structures through source-specific icons and unique coloring schemes, as shown here. Provenance information about these models are clearly provided on the website, and documentation has been created to help users apply the tools available on rcsb.org to study experimental structures and computer structure models. What kind of computer structure models are currently available on rcsb.org? The current set consists of a prepackaged collection of about 1 million computer structure models from AlphaFold DB that include model organism proteomes, global health proteomes, SwissProt sequences, and main sequences. The Rosetta Fold computer structure models from the model archive include a set of about 1,100 heteromeric complexes corresponding to the core eukaryotic protein complexes that were modeled using a combination of Rosetta Fold and AlphaFold 2. These computer structure models collectively cover the entire human proteome and those of model organisms, selected pathogens, and organisms relevant to bioenergy research. During the rest of this course, my colleagues will describe the various features available on rcsb.org that allow users to apply the available tools to study computer structure models alongside experimental structures. Thank you.
Thank you very much, Brenda. If those of you who have questions, please, how much time do we have for questions, Chenghua?
2 minutes.
Do we have a couple of minutes for questions? If you'd put them into the chat, please, I will moderate. There's already been an inquiry in the chat regarding access to the presentations. The link for the PDFs of the presentations will be made available as part of the exit survey. So those of you who fill out the exit survey will be able to get access to the PDFs of all the slides. A recording of the crash course will be uploaded to the Rutgers IQB YouTube channel shortly after the conclusion of today's crash course. Are there any more questions concerning the content of either my very brief introduction or Brinda's excellent presentation?
So I see a question about, do you plan to provide all 214 million AlphaFold models? That's a good question. We are trying to look into it and bring that in some form.
Yeah, so we are going to scale our data delivery capabilities to increase the number of computed structure models that we can deliver alongside the PDB structures. I just want to point out that there was also a question regarding whether or not AlphaFold DB computed structure models are believable, and this question will be addressed by Dr. Chenghua Xiao. I also, just for the other speakers, want to point out that Brinda was coming across being fairly quiet. So if you could, each of you could be sure that you're speaking into the microphones, that would be helpful going forward for all of the participants.
Sorry about that. We didn't realize that during the testing, pre-course testing.
Yeah, not a problem. You were audible, but it would have been easier if we didn't have to turn up the volume. And so then there's a question from Maximilian Pham. Would it be possible for a CSM to be used in a docking program? So the answer to that question, Maximilian, I'll take it, is yes. But I think one has to be very careful about drawing inferences from a combination of small molecule docking to computed structure models. And one has to be very confident that the computed structure model is of high quality and that the results of the docking calculations are very carefully scrutinized. The juxtaposition of these two technologies, both digital, of course, both computational, does carry with it some risks to the practitioners. So we would urge caution in that regard. So Brinda, I think we've reached the end of the questions. Please go ahead with your introduction of our next speaker.
I'm happy to introduce our next speaker, Dr. Shuchismita Datta. Dr. Datta is the Scientific Educational Development Lead and Documentation Lead at RCSB Protein Data Bank. She will describe how to use the rcsb.org search services and the structure summary pages. Over to you, Shuchi.
Thank you, Brinda. I'm going to try and okay, so hello, everyone. We just heard in the last talk that there are over 1 million computer structure models and around 200,000 experimental structures that are now available from the rcsb.org website. So that's a lot of structures, a lot of data. And in order to be able to use this in any meaningful way, the first order of business probably is to find the structure or structures that are related to or relevant to your topic of interest or research project. And then once you've found that structure or those structures, how do you learn or try and understand what this structure is all about? What does it look like? What does it contain, etc.? And then having looked at that, that's what we will cover in looking at the structure summary pages. And then how can using that data help you go forward in your research? We'll try a couple of case studies, and some of these examples will be continued in the subsequent talks. So the first order of the first business that we want to do is to find structures that are related to our topic of interest. And there are a couple of different ways that are available to navigate this large collection of structures and to find things that are relevant and interesting for you. Here we've created an infographic. So I'm going to quickly show you that infographic. This is available on the website, and you will get this link later if you get the PDFs. This describes that there is the most commonly used search option is the top bar search, where on the top of the page, you can type in either the molecule name or any other information that you have about the structure that you want to find. There are also extensive advanced search options that will be discussed in this talk and in the next talk. And then finally, there are these browsing capabilities that we will mention briefly. And then the rest of the infographic actually gives you detailed descriptions of various scenarios. So I encourage you to actually go and look at that and explore it to see what is relevant to your topic of interest. Okay. Now, what I will do is primarily talk about the top bar search and go a little bit into the advanced search options. So just to give you an overview, the top bar search, in addition to typing in a molecule name, a keyword, or a phrase, or specific identifiers, perhaps you are reading a manuscript, and in the manuscript or in the paper, there is a particular identifier listed. You can put in that identifier, or maybe you found these IDs or identifiers in another bioinformatics resource that is relevant to your research project. You can also bring in sequences. So 1-letter code sequences of proteins, nucleic acids can also be put into this top search bar box now. And this mechanism of this toggle switch, turning it on to include computed structure models, the default being exclude, this we will show you how to use it so that you can, depending on what you're looking for and where you want to go with your research, you can either include or exclude CSMs in your search. The other kind of search that is also available right from this top bar search is the documentation search. So this is not going to return structures or ligands or anything like that, but it will tell you if there is documentation, if there are any news items, if there are PDB 101 related articles describing that topic or that term that you have typed in. And we will look at some examples as we go along. The advanced search is not only limited to textual searches, it can actually also, you can use various attributes that are, or metadata associated with these structures. So these metadata for experimental structures, for example, you can search by resolution, you can search by various integrated other metadata, such as enzyme classification or domain classification, or whether or not it is associated with the membrane. These kinds of properties that have been aggregated from other data resources are also available here for performing your searches. You can use sequence-based searches, 3D structure-based searches, and chemical searches, such as using chemical formula, INCI, strings, etc. And so I'm mentioning this briefly. This will be elaborated further in the next talk. Again, here in this panel, notice that there are these toggle switches available to either include or exclude the CSMs. And the browse options currently are only available for experimental structures, the PDB structures. They are not currently available for CSMs, but these actually organize the entire collection of experimental structures according to various categories, such as whether or not it is involved in a particular biological process. Here we are looking at the enzyme classification organization. So there are that many oxidoreductases, transferases, and you can open up these these tree-like things to find specific structures that belong to a particular class. And then that gets you started on your exploration. So now I'm going to actually try a few examples. So we will do some top bar searches, and you're welcome to follow along. So if you are able to go to the browser to rcsb.org, we can try this together. So let's go for it. So I am going to open a new tab. And here, in the top bar search, I'm going to try a simple search for a protein insulin. So I can type insulin. And as I am typing, notice that there are these suggestions that are being made in a box below. So if I ignore these suggestions and just say insulin and press enter, it gave me some 8,300 and something structures, but these may or may not be all just the insulin molecule. And even the first result shows you that this is insulin-like growth factor. This is not what I wanted. So the better thing to do is when I type in insulin, is look through this suggestion and say that, oh, I want structures whose molecule name insulin is matching, not insulin receptor or insulin receptor related protein or insulin degrading, none of those. I want only insulin. So let's try insulin. And these are now all insulin structures from a variety of different organisms. And I can say, okay, I only want to look at human insulin molecules or whatnot. Now, notice that when I did this search, I just used default options, and the default option was CSM off. So there was no CSM included in this search. I can go back and rerun this. Sorry. Insulin. And before I click on enter or do this, I'm going to turn this on. Now, when I run the search, notice that there are 75 computational structures also in this list. And this is ordered in various, so this is a scoring criteria. You can order it in many other ways. But just so you can look at these, here are some of the insulin CSM structures from various organisms. Okay, so with the name, protein name, I can either select from suggestions or just press enter and run this. So I can also come in and say, I have this particular PDB identifier, for example. And so let's try that. So let's say I know that I want to look at this particular structure. So I can put in the PDB ID. And here is the structure summary page for this. I'm actually going to open a new tab and try this other. So this is a CSM structure, sorry. I'm going to skip out so that I can actually copy and paste. So this is an AlphaFold protein identifier. And I can actually paste that in the top search box. But remember, if I run this search with the CSM off, then I will get no results. So I need to turn that button on and run this search. And I find that structure. And I'm going to open that too. So what I did when I found the 1-TRZ and this other AlphaFold protein, I clicked on that and it opened something that we call the structure summary page. And we will look at that in a few moments. But before we do that, I wanted to also point out that other identifiers, such as identifiers from UniProt, that can also be used in the top search box. I can paste this UniProt identifier, and as I'm typing it, it is asking me, is this what you want? And this one, I can choose to run it with or without CSMs and find results. So there is one CSM structure and 300 and something experimental structures that match that UniProt. So in all these cases, we are finding structures that match that particular query. One more example, let's try this sequence search. So this sequence I got from some other structure somewhere else. I can paste that and run a search. And it found all these different matches. Now, effectively, what has happened is when I pasted the sequence up in this top search box, it actually got automatically transferred into this advanced search query option. And you'll hear more about this in the next talk as to how this space, this advanced search query builder, it actually aggregates information from various places and it completes your query so that you can, save this query or run it again, etc. So now we have looked at all these different possibilities for running a search from the top search box. What we haven't looked at is this documentation mode. So let's look at that. So here, there's a pull-down menu where I can change the setting to documentation. And now if I type in insulin, I'm not going to get the insulin structures that I was looking at right now, but there are various documents that describe this. There are news announcements where insulin has been mentioned. There are PDB 101 articles. So this is a completely different collection of results that can help you learn more about a topic of your interest if we have data related to that or information related to that. So I am going to now shift gears to the next topic, which is the structure summary page. So we have run a search, we have found some matches or some structures that we want to explore. If we click on that link and open a structure summary page, this is what the structure summary page looks like. I wanted to give you a quick tour of the structure summary page and then move forward. So on the structure summary page, there is, of course, the identifier, some header information giving you some quality metric of the this particular structure. On the left-hand corner, you are looking at a quick snapshot of what this molecule looks like. And the presence of these different colors tell you that there are multiple chains in this particular structure. Information connecting you to the literature. So the primary article that describes this structure is available from here. The macromolecule section gives you a lot of information about the specific proteins and if nucleic acids are present in the structure, things about that. And if we continue scrolling down further, there is information about small molecules. So this particular structure happens to have these specific ions. And if I click here, I can actually explore the environment around where this ion is located in the structure. And then more details about the experiment and the validation and in entry history. So this is sort of what all structure summary pages contain. These are experimental structures. Now let's look at the structure summary page of a CSM structure. So these structures, if you look at this is not as detailed or informative because there are no ligands, etc. But again, it has the same kind of layout. So in the top left, there is a quick snapshot of what this molecule looks like, and it is colored by that coloring scheme that is explained here. So dark blue or cyan colored regions are highly confident or confidently predicted. The orange and yellow regions are less confidently predicted. And we'll hear more about this in some of the upcoming talks. This gives you a sort of overall estimate of how many amino acids in this particular structure are in the high confidence or middle confidence or low confidence regions, just so you get a sense of what you should do with the structure. Do you want to use this to further explore more ideas? But then below that, there is this macromolecule section that tells you various types of information about the protein that you are whose structure you're looking at, and some
5 minutes.
Yes. Okay, so now that we've looked at the various structure summary pages, we have I'm going to take you through an example where I wanted to show you, like, okay, so you've looked at the structure summary page. So what can you do with this information? So for that, I'm going to take you to one particular structure, 1IEP. 1IEP. Now, this structure is that of an Abl kinase protein bound to a drug called imatinib or Gleevec, commonly known as. Now, in this particular structure, clearly there is the protein. So here we have information about the kinase. And then there is information here about this ligand, the drug Gleevec. The question that I'm going to ask here is that this particular kinase, does it bind to other drugs or other molecules? How would I figure that out? And so to do that, if I click here, this is a UniProt ID-based search that will pull up all other instances of structures in the PDB that have this particular UniProt ID. So if I click on this, let's see what happens. And here I can already see that, oh, there are these other, so it's the same Abl kinase, but it is bound to other different things. So this was our query. And here are different drugs or other experimental molecules that are bound to that same protein. So this can give us some insights and help us define the next question. What we can also do is ask a different question now. So now I'm looking at the drug, Gleevec. And if I ask the question, does Gleevec bind to only this Abl kinase, or does it bind to other proteins too? And I can run a query on that. And if I just scroll through this, so of course there is Abl kinase, but now we are seeing some other kinases show up. And so this way we can actually, so here Sark and Abl, so there are other things that we can find. So this way we can explore the archive starting from the structure summary page and find interesting pieces of information that may be relevant to our research. Now I'm going to do 2 quick case studies, and that will help you see how having access to CSMs can be valuable in this rcsbpdb.org. So for that, I'm going to go to this PDB entry 1B54. So let's go to 1B54. And the question that I'm asking here is, so this particular structure is that of a yeast hypothetical protein. It has this PLP, a ligand bound to this protein, and it is a model for a human protein. So the question I'm asking is that what is that human protein for which this is a model? So I can do a sequence-based search now. And I'm going to say, I don't know exactly how similar the yeast and the human proteins are. So I'm going to be very generous and look at the, let's say the 40% sequence alignment. And what I didn't do is I didn't activate the CSM. So I'm going to do that now and rerun that search. And so now notice that the result set has this yeast CSM and many other CSMs as shown by this listing. I am interested in the human protein. So I'm going to open this up and look for the human protein and narrow that down. And so this is the human protein that the experimental structure was a model for. And if I compare this structure with that 1B54, I can learn how similar or different these 2 proteins are. And maybe it'll give me some ideas about what experiments to perform to understand the function or the structural stability of this particular protein. Okay, and this will be revisited in one of the later talks. And the last case study that I wanted to do is take you to one other PDB entry. This is 3CBB. Let's go to that. 3CBB. Now this is the DNA binding domain of the hepatocyte nuclear factor 4 alpha. This is a diabetes-related gene product. And what I'm seeing in the structure is just the DNA binding domain bound to DNA. But I want to see what else the protein contains, where are the other domains, and can I find more information about this? And to do that, what I'm going to do is I'm going to explore this PDB from this structure summary page, this UniProt entry. And if I click on this, I'm going to open up what we call the protein feature view. And here, this top row is an AlphaFold structure of the complete sequence. And then this was our query. This is just the DNA binding domain. And these are examples of the ligand binding domain, the other part of the protein. And so interestingly here, we see one structure that encompasses both the DNA and the ligand binding domains. So we can look at all these, compare them, and make sense of where these different domains are, how are they interacting with each other, how are they interacting with their target molecules, the DNA, etc. And that can help us design or develop new experiments and progress our research. So with that, I think that was our last case. And I just wanted to quickly summarize that we looked at the top bar search, advanced search, browse options. We looked at a couple of query by example scenarios. And we did these 2 case studies, and some of these examples will be used again in some of the following talks. I'm going to stop here, and any questions?
I think we ran a little bit. behind schedule.
Okay,
So there is a question about a CSM page shows the sequence and rows for PLDDT and disorder. How is that disorder annotation made? Is it from AlphaFold PLDDT?
Yeah, as far as I'm aware, that the PLDDT information is coming from the source where the model was calculated. So we are not computing it, we are getting it from the source. Does that, I hope that answered the question. Brinda, is that correct?
Yeah, I think the disorder, the specific question is how is the disorder annotation made? Not just the PLDDT.
Oh, that's, I think Joanne will be able to address that question far better. And so that's a great opportunity for me to introduce our next speaker. So Dr. Joanne Segura is our scientific software developer for RCSB PDB at UC San Diego. He will talk about more of the advanced search options. He will talk about organizing your results and creating groups, looking at group summary pages, and also 1D, 3D interactive views of these structures. And over to you, Joanne. I'm going to stop sharing.
Thank you very much, Suchi. So regarding that question, how disorder is calculated, we use methods that are integrated in the BioJava library. In particular, I think one of them is iSpread, I think it's called iSpread2. I don't remember exactly the name, but I am completely sure that those methods come with the BioJava library and are computed using the sequences that are found in the structures.
Yeah, thank you, Joanne. And I just want to say, just for the record, that there is evidence that the AlphaFold 2 PLDDT is a superior tool with which to identify low complexity regions of proteins and hence regions of intrinsic disorder. I'm thinking about the work of Rohit Papu and colleagues as a surrogate for disorder. That is not what we're talking about when we, with the disorder predictors that Joanne just referred to.
Yeah, thank you, Stephen. Okay, so let's go to our next section. So in this section, I'm going to talk about advanced search. I'm going to show you how to build your queries to find the proteins that you are interested in. I will show you what to do in the case that you get really long list of results, what clustering strategies do we provide, and finally the 3D view, a really useful tool to explore sequence annotations and map them on the 3D structure of macromolecules. Okay, so first, what is advanced search? So structural data comes with a large collection of metadata. For example, you could be interested in finding these assemblies that has a molecular weight greater than a certain threshold. You could be interested in finding structures that contains a protein with a chain of a particular length, or you could be interested in finding high-resolution crystallographic structures. So advanced search will allow to combine all these different attributes, but even more because we also integrate many external resources. So for example, RCSB PDB data is annotated with protein classification domain databases, as for example, CATH, SCOP, PFAM. We integrate UniProt annotations, gene ontology terms. You could be interested in finding all the structures related to a particular cell process or a metabolic pathway. And also, we provide different types of scientific searches. So for example, sequence identity, finding those structures containing a sequence that is similar to my sequence, or a structure similarity search. So we provide a tool that is the query builder that will allow you to combine all different types of searches into a single query. I will show you also, once you get the results, what operation can you do with them. So we will talk about the search refinement tool. No? This is going to be a really useful utility once you have your result to add new conditions to your initial query. I will talk about what different ranking strategies to sort your results are provided. And finally, in those cases where you have a list of thousands and thousands of results that cannot be inspected manually, we also provide different cluster strategies to try to reduce redundancy and method to explore groups of proteins. No, these proteins that has been clustered together and under a certain condition. Okay, so the advanced search query builder. So as I said, the advanced query builder, what does is merging all these different 8 services into a single query. I'm not going to go into deep detail, but I would like to just to give a brief description of each of them. So we have a full text service. This is a free text, a Google-like. So type your text and we will do our best. We have a structure attributes. As I said, a structure information comes with a long collection of metadata. You would be able to build your Boolean queries, adding conditions on molecular weight, sequence length, and also including these external resources, cat domain, a particular pathway identifier. So similar to structure attributes, we also provide the chemical attributes search. So in that case, is attributes related with the chemical, the small ligands that can be found within the structures. You could be interested in finding, give me all the structures that contains, for example, ATP. Sequence similarity, I am completely sure that most of you have used the BLAST search. No? So in this case, we are not using BLAST. It's a much optimized algorithm called MMSEX. And what it will do is just finding similar sequences to a given one. The structure motif search, I don't want to go into detail what a regular expression is, but the sequence motif I am talking about. The sequence motif will allow you to find these local sequence patterns. Let's say that I am interested in finding all the structures that contains a sequence with a methionine, followed by a methionine, any other residues, and finally a glycine. No? So the sequence motif search will allow you to define this local sequence pattern and find those sequences. A structure similarity, so this is a search based on 3-dimensional shape recognition. So it will search for structures similar in the 3-dimensional conformation to a given one. Finally, we have two more. A structure motif is similar to the sequence motif, but in that case, the motifs are defined as geometric patterns. So 3-dimensional local pattern defined by the selected residues. And finally, the chemical similarity that will allow you to find those structures containing ligands similar to a given chemical smile, or also we provide a sketching tool to input your the 2-dimensional graph of your molecule. So advanced search also allows to configure a bit deeper what you want to get. You can see here on the bottom different options. So let's see what these options can be used for. First of all, the return type. So this will allow you to choose what type of objects do you want to get. No, structures, you will get experimental PDB or computational structure model entries. Polymer entities will return all the distinct chains, proteins or nucleic acids that are found in the different structures. Assembly will return the macromolecular quaternary structure, sometimes also called the biological unit. So in many cases, the structure will not be the conformation of the native complex, but to build the complex, some transformations are needed to be applied to the chains contained in the structures. And in other cases, one structure will contain different assemblies as a combination of a subset of chains. Non-polymer entities will return all the chemical small ligands that are found in the different structures. And finally, the molecular definition, so the chemical components that compose the different macromolecules of the structure. No, in that case, in most situations, you will be able to find the 20 natural amino acids, non-standard amino acids, nucleic acids, and other chemicals that are found, ligands, small ligands that are found in the structure. Okay, I have been talking about what to do when we get a really long collection of results that cannot be explored manually. So for that, we provide two different clusters strategies. One's based on sequence identity that will be clustering your results based on the sequence identity threshold provided, and the other based on uniprot accession aggregation. So in that case, groups, what will happen is that all the members of the groups, all the proteins, will share the same uniprot accession. And finally, in many situations, we will not be interested in exploring single proteins, but family of proteins that are related together with the given condition. So for that reason, we provide also these two options. Once we have grouped the results, we can do, okay, so I am only interested in having a non-redundant set of results. So I am only interested in displaying one representative, one protein for each single group. Or in some other situation, no, I am interested in knowing what is going on. I need an overview, not only of my protein of interest, but also on the related proteins. So you will be able to display the results as groups of proteins and not as single items. And finally, we have here the switch to include or exclude computational models. Okay, so okay, here we are. So this is how a search result page would look like. Here, what is most important, remember, the navy blue color Elden-Meyer flask means this is experimental information, and the cyan color screen icon means is computed structure models. So we get here different items, as I said, by default, so we can use different scoring strategies to sort the results. By default, the order depends on the search score, and this depends on the type of services that have been used. So for example, if we are searching by sequence similarity, results will be sorted by sequence identity scores. But we provide on the top right side of the result list, we have this drop-down sort by that can be used to select the different ranking strategies. One might be interesting. No, I want to sort that by release date or by resolution. I want to see first X-ray crystallography with better resolution and then the others. Or I could be interested in computational models and sort them by the local scores, etc. So many other different ranking strategies. Also on the left side, what you can see here is what we call the refinement tool. So this will allow you to filter the results based on the attribute value selected. No? So for example, if we focus on the first two different checkboxes, it says a structure determination methodology, experimental 2, computational 1. That means that in my result list, there are 2 experimental items and 1 computational. I could click on experimental and only obtain the results, including the experimental condition. So I would remove from there the computational structure models. Also, I was talking about grouping. Of course, that this is not a good example because it contains only three items and can be inspected manually. However, what could I do if I had 3,000? So we could use clustering. So in this case, these three structures contains only a single protein in each of them, and all the proteins is the same, at least from the uniprot point of view. They share the same uniprot accession. So what would happen if I group them by uniprot? That my result list will become a single item. On the left side, I am seeing what I will get if I choose to display the representative. So I have a single group and I'm displaying a structure for each single group. So one group, one structure. And at the right side, all three structures say the same uniprot accession. So all of them has been grouped together into a single group. Okay, so as I said, groups are explorable. Similar to the structure summary page, we also provide the group summary page. And group summary pages would look something like that. So for example, this is a group summary page based on UniProt. So on the left side, you will see a carousel that will allow you to explore the different members of the group. Here you can see member 1 of 3. Remember, this is a group of size 3. We provide the UniProt name, UniProt IDs, here we have the UniProt description of the protein, and we have now different histograms. So these histograms, what display are the distribution of the different group members' attributes, because now I cannot display a release date because this is a group, I have 3 proteins, but what I can do is display a histogram of the release dates. I will show you this when we go to the live demo. And on the right side, what we can see is what we call the group sequence page. So in that case, as it is a UniProt-based group, what we can see are on the top of the protein feature view, we can see the UniProt annotations, and at the bottom, the multiple sequence alignment of the group members. So those that contains this color code scheme, it's because they are computed structure models. And we are showing in these orange to deep blue colors the local score. So the blue regions are those that has a good local score, a good prediction, and those in yellow orange are those that are not that good or can be considered as unobserved regions. So what I think this view can be quite useful because it gives you an overview of the relationship between the local scores and the alignment of the experimental structures. Okay, the most important thing, how to go to advanced search. So each of the pages contains this link that is on the top of the page, just under the main search tool. So you could see the link advanced search. So clicking there, it will navigate to our advanced search page. However, what I find most interesting are those links that can be found in the structure summary pages. So our structure summary pages are really well connected with the other residues. So for example, when I go to the macromolecular section, I can go to the advanced search and find sequence other structures that contain sequence similar to the one that I am interested. The same with three-dimensional structure search. No? Also, we provide, for example, search by UniProt. Give me all the structures that contains a protein that shares this UniProt here. And we have many more links. Now we have, for example, organism. We'll also go to the advanced search and search by all Homo sapiens structures, in that case, gene name. And as we provide links to the advanced search, we also provide links that goes straight forward to the group summary pages. So in that case, all these group summary pages, what has in common is that all of them contains this protein here, these computed structure models. Okay, so finally, I would like to also show what is the 1D 3D view. So this view, what will provide you is an easy way to explore positional features from the 1D on the 3D. To access that, you can do it from any structure summary page, always in the carousel, in the section where it says 3D view, you always will find this 1D 3D view. Okay, so let's go to see all these tools that I have been showing on our website. Okay, so first of all, the main link, advanced search. I click here, I go to our advanced search page. This is our query builder. As I said, this can be used to merge all different type of searches. For example, I could be interested in a sequence similarity search. I can go here in this text box, just type my sequence and go for search. Or here we have our free text. Type whatever you want and it will it will run this free text search for you. Okay, so let's now inspect the different links that I was explaining before. So for example, we have here the structure similarity search, proteins by UniProt. So in this example, what I would like to do is search other structures that contains a chain that is similar to 30% to our protein, in that case, the NOVA2 protein. So when I click here, we can see that it browses to our advanced search. Okay, before going into the results, let's see what has happened with the query builder. So as we can see, clicking there, what has done is fill the sequence similarity search with the actual sequence of our NOVA2 protein. And what we are seeing here are, remember, cyan screen, computed structure models, and Elden-Meyer flask, these navy blue experimental structures. Okay, as I said, the default order depends on the type of search service that has been used. In this case, it was a sequence identity. So if we check what are the sequence identity values, 100%, 98%, 78%, so it decreases because this is the default search for the service. In case that you use multiple services, what the advanced search will do is merging all these scores into a single one. As I said, we have all these ranking strategies. For example, look, I am not that interested in computed models, so I would rather see experimental structure first. So we have here a priority ranking, experimental first, and what now we will get in the first page is only experimental structures. And to find the computer structure models, we have to go to the last pages. Okay, so now refinement tool. As I said, we can now refine this initial query, no, that was based on sequence identity. So in my case, I could be, look, my project is about Homo sapiens, so I am going to be interested in only Homo sapiens protein. So we can just click there and refresh our search, and now what we will get is just the subset of our initial search, only containing, as you can see here, organism, Homo sapiens, organism, Homo sapiens, organism, Homo sapiens. However, refinement tool is a very nice utility because it is quite easy, quite fast, and being able to see the number of proteins, the number of structures that you will get is really useful, but not everything can be done. So imagine that I want to compare now good high resolution structures with computed models. So one could be tempted to go, okay, so let's choose all these ranges of resolution, let's say up to 3 Armstrong, and run the query. So pay attention here, I have 24 experimental, 7 computed models, and run this search. And let's see what happens here. Oops, I have lost my computed models, and this is because the computed structure models, they do not contain resolution information. It has no sense in that context. So as they don't contain, they do not appear here. So for that, to write these more complex queries, we need to use the structure attribute search. And look what has been happening here. I have not talked about that, but each time that we are adding condition from the refinement panel, the advanced search query builder, what does is include those attributes as part of the structure attribute search. Okay, let's go back when we didn't include this refinement resolution, and let's see how we could do that. Another really cool utility is the query history that will allow you to navigate among all the queries that you have done. So it's quite easy now to go backwards, and here we are again, only with my sequence identity condition and my Homo sapiens condition. So what I was saying, I want to include computed models and experimental structures with a resolution lower than 3. So what I can do is, okay, let's add, sorry about that, this is not going to be enough. Let's add a new subquery where I will say determination methodology is computational. Let's add a new attribute, or refinement resolution lower than 3 angstroms. Let's run the query. And here we are. Now we have recovered these 7 computed structure models, and all these experimental, I know that are coming from X-ray diffraction with resolution lower than 3 angstrom, 194, 2.3, 2.4, 2, etc. Okay, let's talk. I know that this is not the best example, because one could say, oh, these are only 25 items, I can explore them manually, but just for the sake of explaining what is the grouping tool. Let's apply grouping here. And let's say, okay, this list is too large. I want to see only those proteins that has a different uniprot accession. So because this is what I'm interested in, I'm not interested in that large amount of result. I want to remove some redundancy here. So I can run the search now grouped by uniprot accession, displaying a representative. So now I am getting only 11 search items, and this is because those are the unique uniprot accessions that are found in my initial search. So this is a representative for a group. So behind these different items, there are other proteins, and only the representative is displayed here. If we were interested in the situation where you are not that interested in a single protein, but exploring or studying a family of proteins, what you could do is display results by groups. Of course, that we will get the same number of items, but in that case, each of these items represent a set of proteins, a group of proteins. And as I showed you before in the slices, let's see how the group summary page look like.
Excuse me, John, 5 more minutes.
Yeah, sure. So here we are in the group summary page. As I see, as I explained, the carousel displaying the different group members. We have these different histograms, and it's quite useful. Histograms behaves quite similar to the refinement tool. So when I am here, I could be interested in, oh, okay, so I am only interested in X-ray diffraction members of this group. So I can click here, and now all histograms will be updated, and the blue portion will be reflecting what is the distribution under my condition, in that case, X-ray diffraction, and the gray are the rest of the protein. So look, for example, here, when I have selected the X-ray diffraction member, the computational has disappeared to 0. Okay, so let's go to inspect the full group, and let's go to the group sequence page. Yeah, as I said, those are annotations coming from uniprot, modified residue, some domain information, and here we have the group multiple sequence alignment. So if I zoom in, I could go to sequence level and really check that this is a real alignment. No, so and what I think is most important or more useful here is that I have the relationship between those regions that can be, let's say, trusted from the computed structure models. So for example, this region here, and I have also the experimental structures that covers those regions. Okay, so finally, really fast, because I am running out of time, let's go to see the 1D 3D view. As I said, in any summary page, 3D view, you will find the 1D 3D view here, and it will open this really cool view where you could see all different types of positional features. We have the secondary structure, we have predicted disorder, we have here the PLDDT values, and of course that this is dynamic. No, I can click on one feature and it will be highlighted on the 3D structure in the viewer. No, so I think in some situations this can be really useful because it allows you to have an overview of all the annotations. So for example, here we can see that coming from a completely independent method of prediction, that the regions that has not been well predicted or cannot be predicted with the computed method also matches with what other methods predicts that disorder is happening. So yeah, this was my presentation. So thank you very much for your attention, and I will be really happy to answer any question.
Thank you, Joan. Please put any questions you have for Joan into the chat, and we'll be happy to respond to them.
So Joanne, there were a couple of questions as you were speaking. Kavita asked, can she search for modified residues in the sequence motif like phosphoserine?
I think Sebastian might
It's
be
only
a better
canonical
person. Yeah,
amino
this is what
acids,
I was
so
thinking.
like anything that is a modified serine will identify as a serine at the end of the day. So
Yeah,
but
I thought so. Thank you.
interesting to know that people are interested in things like that. Yeah.
Right, and then there's an answer from Francisco Murphy, a question from Francisco Murphy Perez concerning experimental features containing something about crystallization conditions.
For the group summary, no, not really. We don't include that that type of information. However, if you think this can be useful, of course that we are open to listen any opinions, because we didn't think about that, and maybe we were missing something that will enrich our group summary pages. So we'll be quite happy to hear your opinion.
Thank you, Joan. And Peter Stogios asks, is it possible to submit a structure to do a search of the PDB, not using an existing structure, but my own PDB file? The answer to that question, Peter, is no. And we recommend that you use something like the DALI server to do those searches. Again, as Joan indicated, if there is a strong interest among rcsb.org users for that functionality, please make it known to us so that we can evaluate that alongside other opportunities for doing improvements.
I think a flavor of that may actually be possible, and this will come up in Sebastian's talk later.
Okay.
Where you can upload files and do some level.
You can certainly upload files and do binary comparisons, but doing a PDB-wide search, I think
motif
is still,
searches are possible.
Yeah, I think it's still the purview of something like DALI.
Yeah. Okay.
Okay, so I would like to introduce next speaker, Dr. Chenghua. He is a PDB developer and annotator, PhD in biophysics and master in statistics, and he will talk about confident measures of computed structure models integrated into PDB.
Thank you, John. So the question was already asked in earlier that about quality of the predictions. Basically, we want to know how much confidence can we have for the predicted models, and if for a big structure, like which part can we trust? To answer those questions, I will first talk about the confidence measures for a computed structure model. Then I'll talk about its applications, especially at rcsb.org. And I will briefly talk about the comparison between the CSM confidence and PDB structure confidence, and finally give a summary of what to trust or not in the CSM. So why did AlphaFold 2 become so popular in the past 2 years? It was actually because there are high accuracy or high confidence. On the right side is the comparison of different algorithms for CASP14, a blind contest for prediction algorithms. As you can see that the dark blue is AlphaFold prediction that has accuracy, the median accuracy on the backbone between the CSM and ground truth reference structures below 1 angstrom, where the others are at a level about 3 angstroms. So the big margin of the accuracy is concluded by that sentence in their publication that AlphaFold 2 is an approach capable of predicting protein structures to near experimental accuracy. Here in the publication and in my later slides, the terms of accuracy and confidence are kind of interchangeably used, although statistically they're different terms. So accurate means high confidence and vice versa. To measure the confidence of CSM, the conventional way needs reference structures. There are 2 ways to do it, and this is actually what CASP is doing. First, you can simply superimpose a CSM to the template, then calculate the either root mean square deviation, also called RMSD, or the template modeling score, TM score. You can also do the superimposition-free distance test, such as local distance difference test, LDDT, which I will talk about in the next slide. But what if when there's no reference structure available? So AlphaFold introduced this predicted confidence of PTM, that is predicted TM score, the PLDDT is a predicted LDDT, and the PAE, which is the predicted distance average for residual pairs. I will focus on the PLDDT because it's the primary per residue confidence measures used in the distributed AlphaFold 2 models. To understand PLDDT, we must understand LDDT first. The motivation to develop a measure such as LDDT can be illustrated on this figure on the right. In this case, the gray structure is a CASP target reference, where the colored one are the CSMs. As you can see, for the conformer A, although the domain structure are quite accurate, you cannot really superpose the CSM upon the target reference for both domains. So to overcome this problem, all atom pairs in the reference structures at a distance closer than the threshold define a local distance set L. And the distance is considered preserved in the CSM if it's within a tolerance same as the one in the L. And for a given tolerance, the fraction of the preserved distance is calculated, and LDDT is simply the average of the fraction at different thresholds. Different tolerances. In fact, the PLDDT is the predicted LDDT C alpha, which means it's on the C alpha carbon only. Therefore, by design, it only reflects the mentioned confidence. And it cannot be considered as an absolute measurement of saturation conformation accuracy. The prediction of PLDDT is in a structure module in the AlphaFold pipeline, later module. And AlphaFold first construct a large training set between the true LDDT between the AlphaFold CSM and the PDB template, where the templates are available. And based on this training set, an algorithm is trained and developed and executed to predict PLDDT based on the predicted model. I want to emphasize that the PLDDT is a predicted confidence. It's not a conventional way of statistical prediction confidence. It's like one more step further. You have a model, and you have the prediction confidence based on the model, and then you calculate the difference between the prediction confidence and ground truth. Then from those to predict the predicted confidence, such as PLDDT. So what the PLDDT score is good score. AlphaFold categorize them into 4 ranges of about 90, between 70 and 90, 50 and 70, and below 50. The overall range is 0 to 100. And each color is actually assigned to the different ranges. And in this case, RNA binding protein NOVA2, each residue is colored based on these 4 categories. The blue color means very high accuracy, which PLDTT about 90 means the backbone conformation is accurate. And the section rotamers are relatively accurate. And the cyan color region means the backbone is accurate, but section conformation is not as accurate. And for the color of yellow, it means the backbone conformation is not that accurate, but may hold some truth. And orange basically means do not trust. So if we focus on one domain of this structure and compare it to the PDB ground truth, we can see that for the blue region, the PLDTT about 90, the structure is identical to the PDB ground truth. And for the cyan region, between 70 and 90, they're similar. But for the yellow and orange region, the structures between the CSM and PDB ground truth is different. Then what about the domain-domain packing? On the left side is again the overall full-length NOVA2 protein based on a PLDTT coloring. On the right side is a 2D array of PAE, predicted aligned error. It's constructed in a way that the 2D array has both X and Y axis for residues from N to C terminals. And each pixel represents a residual pair XY. And in this plot, the color of the pixel depends on the prediction distance error between the 2 residues, XY. And the darker color means smaller error, and light color means bigger error. As you can see from here, this NOVA2 has 3 domains actually. The KH1 and KH2 domain are kind of close to each other, and there's a KH3 domain at the C terminal. And from this plot, you can see that within the KH1 and the KH2 domain, and the distance error is small, which means they're accurate. Even between the KH1 and KH2 domain, for these boxes, the color is still dark, which means the packing between the 2 domains is accurate, relatively accurate. And also within the KH3 domain, it's accurate. However, the packing between, for example, KH2 and KH3 here is light, which means the packing between the 2 domains cannot be trusted. As you can see, the color here for the loops between the domains are either yellow or orange, which means they are of low accuracy or low confidence. And this feature is not yet in RCSB .org, but it will be implemented soon. Now, even though for the domain structure, if the PLDTT is low, we still cannot trust it. On this slide, on the right side, is the PDB ground truth structure that has colors based on the experimental support confidence. And blue means they are confident, and cyan means quite confident. As you can see from it, this structure is very well resolved. However, the prediction is something totally different. And we can safely say that the prediction is not right.
Zhenghua, you have 5 minutes.
Thank you. The last example, I want to point out that the CSM is a prediction on isolated proteins. But as we know, when we do experiments, we use a protein usually in a cellular contact, which means we rarely handle isolated protein. in the cell, and the urea bandwidth is the partner. In this slide, on the right side is this fragment binds to its target protein, and the left side is the predicted model based on PLDT color. As you can see, this fragment, this orange fragment, is basically means, as we said, orange means do not trust, and it means it's basically a random coil or disorder structure, whatever we call it. However, when it binds to its target, it has well conserved loop structure, even with some secondary structure components. So be careful when you use the predicted model in the cellular context. I want to go briefly to compare the CSM confidence versus PDB structure confidence. And as AlphaFold claimed, its approach capable of predicting protein structure to near experimental accuracy, which means they are not as good as experimental accuracy. And you can see from here, the typical PDB structure has a precision of about 0.2 angstrom. And then we see from the beginning of our my talk, the alignment error for the AlphaFold CSM is about 1 angstrom. And also remember, the PDB structure is based on experiments. It's to describe the truth. Well, the CSM has no such property. It's unobserved prediction based on the overall PDB structure and the sequence. We can say that the CSM are comparable to a low resolution PDB structures. And the take-home message is use PDB structure at higher resolution whenever possible instead of CSM. At last, I want to summarize what to trust or not in CSM. So as we talked from me and from other speakers, the CSM is quite accurate at the single domain models of conformational rigid protein. And the backbone conformation can be trusted if the PLDT is high. However, you have to be careful when you're starting domain-domain interactions, as we see from the example, and it complex with binding targets. And if it's conformational dynamics because of either ligand or event-induced conformational change. And we know the PLDT is basically PLDT C alpha only. So the saturation conformation cannot be taken for granted. In fact, even for the best PLDT category, when PLDT is about 90, the first C beta rotamer is only 80% accurate, which means it was within 40 degrees of the correct PDB ground truth. And if you go to C beta, C gamma, the error will get bigger and bigger. I want to emphasize again the message that if a high resolution PDB structure is available, could you use that structure instead of CSMs? And this concludes my talk, and I'm happy to answer any questions.
Thank you very much, Zhenghua. There's a question from Saida Froze. If a PDB structure is not available, is it okay for us to use an AlphaFold structure, meaning a computed structure model? I'm assuming even if the computed structure model has low confidence. We do not recommend using computed structure models that are largely yellow or orange in terms of their prediction confidence, their PLDT. What we see for many eukaryotic proteins is that they look like beads on a string. You've seen this earlier in multiple presentations. The globular portions of the structure that are of the computed structure model that are colored blue and thank you very much, Zhenghua, that are colored blue and cyan are trustworthy. But the other segments of the polypeptide chain, those that are color-coded yellow and orange, indicating low prediction confidence, should not be trusted. Now, there are two ways to look at this. You can be disappointed or frustrated, or you can, as a non-structural biologist, you can seize the opportunity to use an alternative method to try to identify a function or functions for those segments of the polypeptide chain that cannot be characterized using X-ray crystallography or one of the other methods supported by the PDB. So that's a philosophical answer, but one I think that will stand the participants of this workshop in good stead if they keep it in mind going forward. Are there any other questions stimulated by Joanne's talk?
There is another question. Is it possible to find intrinsic disorder protein with a CSM that is in the confidence region? Stephen already indicated that there are people doing researches indicating that the PLDDT actually is considered a good indicator of to indicate internal disorder protein regions. So typically, a low PLD, as you can see here, these are low PLDT regions, and they are basically random coil or disorder. But I think there are always, sciences like at the front end, there are always cases that are exceptions. And whether or not those exceptional cases are trustworthy, it depends on it's case by case. So that's would be my opinion.
Thank you very much, Chenghua. Those of you who are interested in learning more about the question of discrepancies between the PDB and PLDDT can look at Chenghua's recent paper that appeared in Structure, Xiao et al. It was published online at the end of August, and that reviews some instances where you had confident structure, you had well-resolved structures versus low PLDDT scores and vice versa. So I think we should now move on to our last speaker of the session. We're running a little bit low on time.
Sure.
So
our last,
could you please introduce Sebastian?
Sure. Let me stop share first. Our last speaker is Dr. Sebastian Bitrich. And Sebastian is a PhD in structural bioinformatics, and he's a scientific developer at RCSB.org. He will talk about, as you already see from the titles, the 3D visualization and 3D alignment at RCSB.org.
Yeah, thank you so much for the kind introduction. So yeah, I want to talk about visualization techniques that you can find on RCSB.org. And to continue a train of thought for the first part, I would like to show you ways how you can color structures by those confidence values. So how you can obtain those PLDDT values that we just saw, and how you can access RSCC values for a subset of experimental structures. And for PLDDT values, it's pretty straightforward for any AlphaFold structure, which is just a single chain. So if you go to the 3D view anywhere on RCSB.org, it will by default be shown in this blue to orange gradient, or you will have this PLDDT coloring theme applied by default. However, I want to point out that in some cases, maybe you're looking at for example, at a Rosetta fold structure, which contains 2 chains. And in that case, we didn't opt to show these PLDDT scores by default, and rather wanted to emphasize the fact that there are multiple chains present in that particular structure. So if you encounter a case like that and you don't see this coloring by default, you can always go to this quality assessment panel, and it will give you an overview over all the applicable coloring themes that are motivated by some form of quality measure. So I think that's a pretty convenient way to, for example, color model archive structures or Rosetta fold structures. And it also works for experimental structures. And to give some more details on those PLDDT coloring themes. So again, we have everything in dark blue basically resembles high confidence regions where the AlphaFold 2 network is confident in its prediction. And we also see, especially in, for example, the N and C terminus or in certain regions, it's quite common that you see lower confidence regions. And if you want to get additional information on any of those residues, you can always hover over that particular residue in this 3D view, and it will bring up this tooltip that you can see in the bottom right corner, which gives you additional information like which residue you're looking at. So that's position 38. You can see the actual PLDDT value, and you can also see a high-level description of what the color means, which might be helpful in an educational setting, for example, or if you're not using this particular color theme every single day. And I also want to point out how you can obtain this coloring theme if you don't have access to the quality assessment panel. For example, if you're looking at the structure alignment, which I will show to you in a couple of minutes, then the way to request those colorings is to go to the components view in Moster. And here you can see I actually loaded 3 structures. And you can go to any entry. So you can see that I loaded a AlphaFold model and I loaded 2 PDB structures in this particular example. And if you want to access this coloring theme, you want to go to those 3 additional dots, which give you options to manipulate this particular component. And in here, you can see that you're looking at a cartoon representation for this AlphaFold structure. And if you click on that, you get another level of options. And in here, you get access to the color theme. So initially, it will be set to something different. But if you click on whatever is in this box, you will see all the color themes that are applicable to this particular structure. So I think this is in general helpful if you want to change the color of something, and gives you access to many nice properties like accessible surface areas, for example. But under validation, you will find PL DDT values, and the same is true for the RSCC values that Zhenghua alluded to. So, and again, Moster or the 3D viewer will basically make this decision what is applicable for whatever structure you're actually looking at. And finally, I would like to point out that you can see this low confidence region in the bottom right corner, where the AlphaFold network can't make a good prediction, and it will place residues somewhere in space, but it's also pointing out that this prediction shouldn't be trusted. And interestingly, if you look at the PDB structure for the same sequence, you can see a similar tendency that overall, if you color it now by those RSCC values, the overall structure quality is quite high, and you have like large regions of confidently modeled residues. However, you have this particular region where you have low quality according or as quantified by those RSCC values. And you can even see that there's a small sequence region surrounding this NINA sequence where the experimental method, we just don't have experimental data for those 5 residues, for example. And I just want to point out that where experimental methods struggle, the AlphaFold network tends to struggle. And also AlphaFold at the end of the day is trained on PDB data. And if you don't have structure data for this particular case, then AlphaFold also struggles to learn any meaningful information. And again, just wanted to make this point that it really makes sense to compare computed structure models with PDB structures where available, and structure alignments are a really powerful tool to make those kind of assessments. And because of that, I want to show you also how to create your own structure alignment. So basically show you how to recreate this view that I just showed you, where you can visualize 1 computed structure model and 2 PDB structures and compare them by structurally aligning them. And to give you some more motivation why you should care about structure alignments, I want to point out that PDB structures are really rich in information. And as we heard in various talks already, oftentimes you can see ligands being present in PDB structures, and that's never the case, at least at the moment, for computed structure models. And also you can see PDB structures occurring in more complex scenarios where they interact with other proteins or where they maybe form biological assemblies. So you get a lot of information that will be present in the form of coordinates that will be deposited in a PDB structure. And it's not the case at the moment for computed structure models. So all of them are kind of information sparse, but still, if you're looking at one particular sequence, you might want to know for a computed structure model, where can a ligand bind? And again, computed structure models can be basically enriched by information coming from PDB structures. And so you can transfer or map information from PDB structures onto computed structure models. It's also computed structure models. We have 1 million of them integrated at this point in time. So it's a large set of data that is difficult to navigate, and structure alignments can help with that. And as shown initially, it's also a tool to make assessments or to understand the limitations of whatever structure you're looking at. So yeah, and because of those, because of the importance of structure alignments, we try to make it as convenient as possible for you to create your own alignments. So there's a dedicated page application on rcsb.org/alignment where you can create your own alignments. And this tool allows you to align one or more chains to a reference chain. So it's basically pairwise, always with respect to one reference, but still you can align 10 structures or 10 chains if that is necessary in your particular use case. And the tool fully supports PDB structures, of course, as well as computed structure models. And you can load individual structures by their PDB ID, by their computed structure model ID. And you can even point to external structures. So maybe there's something in AlphaFoldDB or any other resource that isn't yet available on rcsb.org. You can point to those structures, and you can even upload your own MMZIF files. So maybe you're running a molecular dynamic simulation and you have one frame of your trajectory that you're particularly interested in. You can upload that and create alignments based on that. And yet this tool provides a number of options, and it allows you to visualize everything directly in Monster, and you can even download the aligned structures or some meta information, like what's the transformation matrix that you would like to, or that you would need to apply to one structure to align it onto the other structures. And just to give you an overview over how the input form looks like. So basically you can punch in individual computed structure model IDs or PDB IDs, and you want to select one chain that you want to align, but it's as easy as that pretty much. You can add more structures using this plus icon, and this dropdown allows you or gives you access to the other input options to point to a URL or to upload your own file. And if we circle back to this mapping idea of taking interesting information from a PDB structure and mapping that onto a computed structure model, we can do it with a query like that. And once you have access to the slides, you can also follow this link and revisit this example on your own. So basically the results will look like this, where you get this overview over your alignment with some meta information, and you can see the sequence alignment. And it's already obvious that this is it's a somewhat trivial example where you have perfect sequence identity, for example. So you'd also assume that your alignment is like really good quality. And you can look at the scores for your alignment. So you can see the RMSD value as well as the template modeling score, which ranges from 0 for no correspondence to 1 for perfect correspondence. And as a rule of thumb, anything above 0.5 means that you're looking at the same fault. So this is a really good alignment, is what I'm saying. And again, it's a trivial case with 100% sequence identity, but still you can get some information out of that. So if you look at the 3D visualization, you will see something like this, where you get access to Monster and you will see the result of this alignment operation. And I guess the most important input option here is this select view dropdown. So initially you will only see residues that have a correspondence in the other chains that it was aligned to. But I think the most meaningful option, at least for the question that we try to answer, is to switch to structures where you can see the whole structure with everything else in it aligned to your reference structure. So you can see all the information from those two PDB files aligned to your computed structure model. You get access to the ligands and all the waters that are present in the PDB structures. And yeah, basically you can manipulate the visuals by using those eye icons. So for example, I've hidden all the water molecules because they're somewhat distracting. But if you're looking at the overall alignment, you can see a really strong correspondence between all structures. And you can also make this assumption that the AlphaFold structure, because it's structurally that similar and has a similar binding site, in particular, the ligands that are present in the PDB structures, this gray and the green ligand, that the AlphaFold structure, even though it doesn't explicitly have this ligand present, you can still make this assumption that, yeah, because it's so structurally similar, most likely it's able to bind the ligand in the same fashion as the PDB structures do. And I also want to show you another way to create structure alignments, which are based on structure motifs. So previously we looked at global structure alignments, which try to align the overall structures. But I think that structure motifs, in the sense of a handful of residues and a characteristic orientation, is also really meaningful, because basically structure motifs are a way of reducing the enormous complexity of a protein structure. And most of the time you're only interested in a binding site or an active site. And the rest of the protein structure is just there to fix this binding site in place and to modulate its properties. So it's a nice
Sebastian, apologies for interrupting. You have 5 minutes.
Yeah. And
thank you.
So I think it makes sense to use this representation for more complex scenarios. And you can make use of that by defining structure motifs and searching for them. So we offer tools for that as well. And everything is, again, fully integrated into Molster, where you can define and extract whatever binding site you may be interested in, and you can search for it on rcsb.org. So basically it will look like this, that if you go to a structure summary page, for example, for 1B54, you can create this ligand focus where you focus in on one particular biologically meaningful ligand. And it will look something like this, where, again, you zoom in on the ligand. And once you arrived at this step, Molster will show all the non-covalent interactions that are formed between the protein and this particular ligand. So you can see this network of hydrogen bonds that fixes the ligand in place, or that mediates those interactions between the polymer or protein chain and the ligand. And if you switch to this selection mode by clicking this cursor icon, Molster will allow you to identify or select individual residues. So here I selected those 4 residues that are close to this aromatic ring because I deemed them of interest. And just by selecting those 4 residues, you fill this list of the residues that you're actually interested in, that you want to search for on rcsb.org. And as a final step, you will click Submit Search, and this will allow you to find any occurrence of those 4 residues that have a similar orientation. And results will look like this, where you can see, if you opt in, you can see computed structure models, you can see PDB structures, and you get a lot of meta information that is specific to the structure motif alignment itself. So you can see what were the residues from this particular computed structure model, for example, that were matched to your query motif, and what is the RMSD of this alignment. So what's the alignment quality for those 4 residues that were mapped to the 4 other residues from this structure. And as a last thing, you can click on Align to visualize the alignment in Molster. And it will look something like this, where you can, initially, you will get a visualization, this ball and stick representation for your query motif, what you're searching for. And you can see how the 4 residues that the algorithm identified in the computed structure model in this example, how they are oriented. And you get access, like in many other places in Molster, to this components panel, where you can adjust the visibility of all the other components. So again, initially you only see this ball and stick representation for the computed structure model and the PDB structure motifs, only those 4 residues, but you can enable the visualization of the overall polymer chain of the PDB structure. And you can also activate the visualization of the ligand that is present in this PDB structure. And yeah, the last thing I want to point out is that how similar the the binding site residues are in the blue AlphaFold prediction in comparison to the PDB structure. So I think that's pretty remarkable that at least in this example, you can really see basically a perfect prediction, at least at in this small region of your protein structure. So, and yeah, for the sake of time, I guess I'll come to a close, and yeah, feel free to ask questions, or if we missed something over those 2 hours, then feel free to post again in chat, and I'll hand it back to Stephen.
Thank you very much, Chenghua and Sebastian. Let me just share my screen and make a few concluding remarks. So again, our thanks to those of you from the United States and around the world who joined us today. The opportunities for sending feedback include the crash course survey, which you will all receive this link via email. You can also simply go to the rcsb.org website and click on Contact Us and type in a message, and that will be responded to by Rachel Green or one of my other team members. I wanted to point out to those of you who are excited by what you see at RCSB Protein Data Bank that we are in a growth phase and are recruiting software developers both at Rutgers in New Jersey and at UCSD. We're also looking to recruit postdoctoral researchers both at UCSD and Rutgers. And then finally, we have both undergraduate summer research openings and gap year opportunities for students undergraduate students who are either working on or have completed computer science degrees to come and work at Rutgers with us. And I also want to draw your attention to the fact that on the 10th of November, we will be having another somewhat longer crash course. This is being conducted in collaboration with the Department of Energy Knowledge Base, known as K-Base. And the focus of this particular crash course will be on using a combination of K-Base and PDB data and computed structure models to better understand plant proteins. And our focus is going to be on the proteome of Arabidopsis thaliana, which is the hydrogen atom of plant molecular biology. You can see from the structures and computed structure models arrayed around it using our color scheme that there are both experimental structures in the PDB of Arabidopsis thaliana proteins, and then of course a much larger number of computed structure models of Arabidopsis thaliana proteins, which are being made available on rcsb.org in the form of these CSMs that we have integrated with the PDB data. It now remains for me to thank everyone involved in the RCSB Protein Data Bank. You can see their faces here. It's a huge team effort to deliver this information, to work closely with the structural biology community, to bring new structures into the PDB, to safeguard the archive in our role as the Worldwide Protein Data Bank archive keeper, and to deliver this information to you, and also to do outreach and education on and training on our RCSB PDB introductory website, which is known as pdb101.rcsb.org. I'd specifically like to acknowledge the long-standing contributions made by our late colleague, Dr. John Westbrook, who passed away tragically last year, and to thank all of our speakers today and members of the team who helped with the logistics, Michelle Sangera and Stephen Dodzik. So thank you. Being respectful of your time, we're going to bring this to a close. Please do use the opportunities to provide feedback, either through the SurveyMonkey exit survey or on our Contact Us button. Good luck, be safe, and use both PDB structures and computed structure models with a clear eye as to their quality and their accuracy.